I'm going to share করছি আমি হ্যাঁ হ্যাঁ এবারে এসছে দেখা যাচ্ছে Are you able to see রুচি
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Okay, okay. এটা নাফিসার গলা নাকি লাস্টটা
না স্যার রুচি
রুচি আচ্ছা Okay. Are you people able to see this antibody targets and functions?
Yes, sir.
Okay. So
Yes, sir.
antibody has got the following targets and functions. What are they? First, check complement fixation. So you have read about what are complements. So complements are basically a group of proteins which basically make holes in the 20 plasma proteins which target to kill invading antigens and bacteria. Okay, so first before complement gets fixed, you have several antibodies coating the bacteria to make it more tasty. So we will now talk about exactly how things work. So complement fixation. So complement fixation means first the antibodies coat, then on the FC region, that is the tail region of the antibody, which we had seen, you will find the complement binding, and then there is a complex formed, and then 20 plasma proteins, they work, C3, C4, they all work, and they make a hole inside the bacteria, and the bacteria dies. So this is how antibodies function with the help of complement. So the first you have an immune complex formation exposes a complement binding site on the C region of the immunoglobulin. So what is the C region of the immunoglobulin? This region. And here is the complement binding site. This is the constant region. This is the variable region. So here, first this region is exposed. These antibodies, they come. This is the antigen binding site. On the basis of this site, they bind on the particular pathogen or bacteria. And then this site is exposed. Now the complement will come, complement proteins will come and bind over here. Okay, complement to the C region of the immunoglobulin. Immunoglobulin and antibody are the same thing. Complement fixation results in cell lysis. So the complement binds and the cell is lysed. The next part of antibody is neutralization. The immune complex formation blocks specific sites on virus or toxin and prohibits binding to tissues. So when all the antibodies bind on top of this particular antigen, what it does is that this virus or bacteria, they cannot go and bind to other cells because it has been covered by all other antibodies throughout. It has been coated. Then agglutination. Cells are cross-linked by immune complexes and clumped together. We will see the photo right now. So many of these viruses together, many of these bacteria which have entered inside, they are all clumped together. So they are not moving about freely throughout the body. Then precipitation. Soluble molecules such as toxins are cross-linked. They become insoluble. So like a salt and soluble substance which are soluble in your blood, but they are causing harm. Some bacteria toxin which has been released, some poison which has been released. So they are causing harm. So what they do is the antibodies, they go and bind to them, to these solutes, and what they do, they cross-link them and then they make them insoluble. Once they're insoluble, they cannot go and harm your tissues. And the debris which is formed, inflammation and phagocytosis are stimulated by the debris. Now later we will see this photo a bit, same thing, a bit in a larger manner. এটা দেখতে পাচ্ছ এবারে?
Yes, sir.
Okay.
Yes, sir.
Yes, sir.
Okay, this is the antibody, this is the antigen. The antigen and antibody form the antigen-antibody complex. First thing is, it inactivates the antigen by first, what is it? Neutralization. So this yellow is the antigen and the blue is the antibody. They have covered it. So they are covering it. And then what happens? All these red things are the bad things. What they do? They all agglutinate it. All the antigens are agglutinated. And then soluble antigens, they are held by this antibody and they are precipitated. This antibody, they precipitate out. Then what it does? It fixes the complement. Once the antibody binding, what happens? These complements come and they activate and they fix it. And this leads to cell lysis. And these two things together, they enhance phagocytosis and inflammation. So now antibody targets and function. First, we must talk about what are monoclonal antibodies. Antibodies produced by descendants of a single cell. So when some antibody antigen is injected in your body, you have several antibodies made against that particular antigen. Now this antigen has got lots of shapes. This antigen has got lots of shapes. অনেক shapes রয়েছে. Some are squares, round, triangle. So normally what happens, the response, antibody response is, you have a polyclonal antibody formed. That is, against each shape of one antigen has got, say, 5 shapes. So against those 5 shapes, you have got 5 different antibodies being produced. And the best thing is that against those 5 shapes, each antibody which is being produced is produced by one type of cell. Okay, so basically for that antigen which has got 5 types of shapes, you have 5 different B cells producing 5 different antibodies. Actually, वो एंटीजन का जो 5 शेप है, वो 5 शेप के अगेंस्ट 5 एंटीबॉडी प्रोड्यूस हो रहा है, और 5 एंटीबॉडी प्रोड्यूस हो रहा है 5 बी सेल से, क्योंकि एक बी सेल सिर्फ एक टाइप ऑफ एंटीबॉडी बना सकता है. So each one of them, each B cell is a clone. A monoclonal antibody is an antibody which is against one shape. It is produced from a descendants of a single cell. Say triangle wala shape ke against mein you have got an antibody formed, and that particular cell which is making that antibody against the triangle antigen, that produces many same type of clones of each other. So सिर्फ वो triangle वाला के ही against में वो antibody तैयार होगा. This is known as monoclonal antibody. Normally what happens when some bacteria virus enters, they have got lots of shapes, so the response is a polyclonal response. आगे से पीछे से ऊपर से नीचे से सब जगह पे different type of cells are attacking only one pathogen. But in treatment, it is better if you have an antibody, say one cell से एक clone बना सकते हो, and that is producing an antibody, then it is very useful for many purposes, for medical treatment. Now, for example, say You need a particular antibody against a particular protein, a particular antigen. You need an antibody. Now, you inject a rabbit with that antigen, and then you take the serum from the rabbit, kill the rabbit, and take its serum, and then you use it. Now, then when the serum is finished, what happens? The rabbit is also dead. You have to again make it. But in monoclonal, what happens? If you can isolate that one particular cell which is making that one type of antibody, you can fuse it with a cancer cell. Cancer cells are immortal, they keep on growing. And then in a cell culture, you can keep on growing these cells, and these cells will keep on producing this antibody. It will be secreted. So, एक बार एक if you get one monoclonal antibody producing cell, तुम्हारा लाइफ बन गया. उससे पूरा जिंदगी भर तुम you can sell that antibody and राजा बन जाओगे. Otherwise, every time you have to kill the rabbit, and there is a cost of the rabbit. Otherwise, वो सेल में से सेल को सेल कल्चर ट्यूब में you can grow, you can take that antibody, and then there are many autoimmune diseases like myasthenia brevis and all these things. And what happens that this muscle degeneration starts to take place. So it's an autoimmune disease. So what you do is you get an antibody against these particular antigens which are at self antigens, antibodies which are produced. You make an antibody against the self antigen antibody. And what happens every time you inject that antibody? What happens? A self autoimmune disruption, it stops. So your own antibodies will not destroy your own muscle cells. So you have to keep on buying this antibody and injecting it inside yourself if you have this particular disorder, and then you will stay fit. Like diabetes, you keep on taking insulin throughout your life. Like this, now जिसने बनाया इंसुलिन वो तो अमीर बन गया. ये इंसुलिन बैक्टीरिया में प्रोड्यूस करता है, breaks it, and then they sell it. So similarly, like this particular cell, what happens? It keeps on making this antibody, and you just simply purify that antibody and sell it in the market. And the sick person will buy that antibody and inject himself and will be fit. So एक बार एक monoclonal antibody बना लिया तो राजा. But if it's a polyclonal response, then there is a problem. The problem being that this particular एक बार वो serum खत्म हो गया, बस खत्म. You have to take another rabbit, make a fresh. You don't know if the response, the quality will be as good as the previous one. So this problem is always there. So this is what is written: pure antibody preparations that are specific for a single antigenic determinant. Antigenic determinant is just one shape. Like I told last year, maybe a triangle shape, a square shape. It is one cell is producing an antibody against one particular shape. That shape is the antigen. So it is needed in research, diagnostic, and therapeutic uses. Now, from the humoral response, the antibody response, we are coming to the cell-mediated immune response. That is T cell activation. This involves recognition of plasma membrane surface antigen only. So before we go over here, I want to show a picture. Are you able to see this picture?
Yes, sir.
This is a cytotoxic T cell. This is antibody antigen presentation. This is a T helper cell, a type of T cell, T lymphocytes. And this is a phagocyte, like macrophage or the neutrophil or whatever. This is a phagocyte. What a phagocyte does, it ingests the pathogen inside, it breaks it into small antigenic determinants, and they take MHC molecule and they present it on the surface. So this particular phagocyte is on this particular MHC class 2. There are class 1 and class 2. So a specific MHC class 2 is found on specific immune cells like macrophages, dendritic cells, neutrophils, only on specific immune cells. And MHC class 1 is present on all cells. So all cells, say for example, a cell is infected with a virus or some toxin. What happens? That cell, that particular toxin is expressed, is processed, and that protein is expressed on MHC1 molecules, is displayed. So even if it's a self protein inside your body, say your kidney cell, your kidney has got lots of different of your own proteins. So each of these proteins are expressed by MHC1 molecules. Little parts of this, little antigenic determinants are expressed. Not the whole protein, little, all proteins of your body are expressed on all cells on MHC1 molecules. But most of them are self. When the T cell is being selected, self ones, they are eliminated. So they are not allowed for further propagation. ये याद है तो?
Yes, sir.
Yeah, the self ones are eliminated. So if the MHC expresses self ones, no problem. It is not going to attack that cell. But moment another protein enters from some outside, protein comes out, non-self thing comes inside, some bacteria, virus, or something, some of their proteins comes inside. That means the moment the non-self is, these T cells will recognize it as the infected cell, and these killer T cells, they will come, killer T cells, cytotoxic T cells, they will come and bind, activated cytotoxic T cells, they will come and bind and kill that infected cell with the virus and eliminate the danger threat. So you have MHC2, which are found only on specific immune cells like macrophages and these phagocytes. They present this. So on this MHC2, these T helper cells, they bind. Then these T helper cells, they get activated. They release certain chemicals called cytokines. They activate the cytotoxic T cells. And cytotoxic T cells will bind. Now these T cells have already been activated by this particular MHC2. So these MHC2 T cells are activated and non-self antigens are presented. So a repertoire of different T cells will come, and it will, the T cell which will bind with this class 1, it will bind and it will cause cell death. That infected cell will die. Is it clear? Is this clear?
Yes, sir.
Cornali?
Yes, sir.
Okay. Now after this, I'll get back to the main place where I'm been going. So T cell activation involves recognition of plasma membrane surface antigens only. So on the plasma membrane, what you have the MHC1 or 2 molecules which are displaying the antigen, surface antigen. And this antigen is processed inside, and this little, all even self, non-self सब express होता है, लेकिन self में bind नहीं करता, only non-self में bind करता है. तो non-self जब bind करता है और मार देता है उसको. So antigen is combined with MHC and displayed on plasma membrane. Then you have T cell receptors which bind to the MHC and are stimulated by the associated antigen. So T cell receptor, T cell में T cell receptor आता है, CD4 or CD8. CD4 is in helper cell, CD8 in cytotoxic T cell. CD8 receptors, they recognize MHC with antigen, उसमें bind करता है और मार देता है. फिर कोई को स्टिमुलेटर भी रहता है, लाइक सम साइटोकाइंस, इंटरलीकिन्स, ये क्या करता है, से अगेंस्ट पर्टिकुलर ट्रायंगल वाला एंटीजन के अगेंस्ट में a T cell binds. They also stimulate similar type of clone बना देगा same T cell का. So you are infected with COVID, and so your T cells, one of the T cells recognize COVID and bind to it. So immediate one binding, what happens? Immediately that many of these T cells which bind to COVID will be produced. There are lots of interleukins and stimulators, but in COVID severe cases, the main problem is Before all this activation, T cell, B cell activation takes place, the patient is dying. और इंटरल्यूकिन एंड ऑल दैट इन्फ्लेमेशन होता है, सांस रुक जाता है. तो थोड़ा भी लंग में प्रॉब्लम है या हार्ट में प्रॉब्लम है, दे कैन नॉट टेक द प्रेशर. Even our botany teacher Animesh sir, he's not that old. He was infected with COVID. He had a severe heart chest pain. It was almost like a heart attack. He was taken to hospital, and then the muscles outside his heart got infected, not the heart itself. But if he was his pulse was fluctuating from 200 to 100, then again 200, he was given electric shock on his heart, and then it started pumping. It happened last month only. And finally, his heart had no problem, but since he was something around 40, he survived. If he was 60 or 70, he would not have survived this. Such an old person's heart with comorbidities and diabetes, that person would not have survived. So this was the problem with COVID. But your body has got infection fighting mechanisms. One side you have the B cell, which is this antibody and complement fixation. Second side, you have these T cells which are coming, and these virus infected cells which are or cancer cells. Cancer cells are self non-self का एक बात होता है. They will be expressed, so they will bind to cancer cells and kill them also. So we also got a natural mechanism to fight against cancer. Most of the time, 1000 में से एक बार maybe when it misses, of that person gets cancer. इतना सारा we get so many mutations in our body. इतना सारा mutation होता है. हर बार क्या कैंसर हो जाता है? नहीं होता है. कैंसर जब होता है तो मतलब आफ्टर सो मेनी डिफेंस इट फेल्स, तब कैंसर होता है. फर्स्ट डीएनए रिपेयर होता है, बहुत सारे चीज होता है, दैट सेल इज एलिमिनेटेड. बट सडनली इवन सब कुछ ये होने के बावजूद भी ये चीज हो जाता है, और उसके कारण मर भी जाता है. इंसान मर भी जाता है, तो ये मतलब फेल हो जाता है. बट इवन द इम्यून सिस्टम has got by this T cell mechanism, it can recognize cancerous cells. कुछ अलग रहता है, some cell surface markers होता है कैंसर सेल्स का, तो अगेंस्ट दैट बनता है. So in the absence of a co-stimulator, the T cell becomes tolerant to antigen energy. This is a very very bad thing. Interleukins are a co-stimulator नहीं रहता है. What happens? The T cell will not bind. That T cell will become tolerant to that antigen, and this is a very bad thing. This is known as energy. Now, MHC molecules के बारे में, major histocompatibility complex. So basically found in mouse. It was found that every individual has got a different type of histocompatibility complex. It's a cluster which lies on chromosome huge MHC1 and HLA locus. This gene produces different individuals have got different MHCs. So MHC1 is virtually found on all tissue cells, and MHC2 is displayed on the plasma membrane of only some immune system cells. Okay, MHC1 and MHC2, both of them, they display particular antigen for T cells. Lekin what happens here, phagocytes like macrophages and all, what they do, they first what they do, they take gup karke they take up the pathogen. Then the pathogen is broken down into little pieces, and little protein polypeptide, little antigens are produced, and they are displayed on MHC2 molecules. MHC2 molecules on those phagocytes. They are recognized by a special type of T cells known as T helper cells. These T helper cells, on binding, recognize it, they release co-stimulators like cytokines, interleukins, and these co-stimulators, what they do, they stimulate these co-stimulators, they stimulate the cytotoxic T cells. Now the cytotoxic T cells अब क्या करता है, MHC1 में भी the same if the body is infected, the MHC1 tissues are infected, that is also expressing that particular antigen of that particular virus or bacteria. फिर उसको जाके बाइंड करता है, आर्मी को एक्टिवेट कर दिया, आर्मी को एक्टिवेट करने के बाद आर्मी आके भड़ भड़ करके मार देता है. क्लियर?
Sir.
Okay. So MHC display properties. MHC1 is expressed on virtually all tissue cells. See, antigenic peptide class 1 MHC यहाँ पे है. Actually क्या होता है? First is endogenous antigen. इसको बड़ा करता हूँ इसको. फोटो को इसको बड़ा कर. দেখা যাচ্ছে?
Yes, sir.
Okay, this is endogenous antigen. This is a viral protein which has some virus has infected. This viral protein has come inside. This is degraded by protease. This is processed in class MHC1 inside the endoplasmic reticulum, and class MA. This is expressed on the plasma membrane. Now your T cells will recognize these class 1. TC cells will recognize these class 1 antigen and इस सेल को मार देता है, because this is a virus infected cell. Virus infected cell will, what will it do? It will propagate the virus more and more. This will die. ये मर जाएगा, लेकिन बाकी आसपास के लोगों को बचा देगा. So MHC1 is on virtually all tissue cells. All tissue cells are there, and they are all expressing these particular, all even self anti cells का भी protein they are expressed, but they are not recognized by T cells. HIV में what happens? Your T helper cells, the ones which bind to the phagocytes, they are destroyed. Result is that the T cell response, T cytotoxic cell भी काम नहीं करता है. So half your immune system is destroyed. Not only half is destroyed, even this T helper cells, they stimulate B cells also to secrete antibody. So in HIV actually the problem is there. The T helper cell पे problem होता है. They are compromised. The CD4 receptor. And as a result of it, your immune system is destroyed. और तुम दूसरे किसी इसमें you will die. So what do? They display only proteins produced inside the cell. Endogenous antigen is equal foreign. What are endogenous antigens? They are foreign produced by the cell, like virus, cancer. उसको भी डिस्प्ले करता है. वायरस कैंसर सेल का भी मतलब अंदर ही क्या कैंसर सेल बना कुछ अलग हो गया कि একটু আলাদা হয়ে গেছে, তাকেও ডিসপ্লে করে তাকেও রেকগনাইজ করে মেরে দেবে. সো হোয়াট দে ডু, দে স্টিমুলেট CD8 সেল প্রোডাকশন. What are CD8 cells? CD8 cells are cytotoxic T cells which express the CD8 receptor. CD8 receptor is for killer T cells. Cytotoxic T cells, formerly they were called as T8 cells. MHC2 is found only on plasma membrane of B cells, some T cells, and antigen presenting cells like macrophages, neutrophils. So what do? They help the helper T cells. Helper T cells को क्या करते हैं? They activate the helper T cells and they stimulate these interleukins and cytokines, and these interleukins and cytokines they stimulate the cytotoxic T cells जो मारने वाले लोग को स्टिमुलेट कर देता है. ये प्रचार करने वाले लोग हैं टी हेल्पर सेल्स. पहले ये स्पाइस होता है, देखता है कि एनिमी घुस गया है टेरिटरी के अंदर में, अरे चलो इंफॉर्मेशन रिले कर देता है आर्मी को, आर्मी फिर आके मारता है. लेकिन स्पाई लोग मारता नहीं है, स्पाई लोग इंफॉर्मेशन रिले कर देता है. तो टी सेल हेल्पर सेल्स आर लाइक स्पाइस. वो लोग इंफॉर्मेशन लेता है कि ओके सम कोई गड़बड़ है, and it is expressed. They relay information by the help of cytokines and interleukins, and they essentially activate the cytotoxic cells, even B cells also, वो भी antibody release करता है और भी. And then, so this is a link between humoral and cell mediated response. उसका link क्या होता है? Your T helper cells releasing interleukins, which are stimulating B cells to release antibodies, and ये हो गया humoral पे link, और cytotoxic का link हो गया, it is stimulating your cytotoxic T cell. तो ये हो गया cell mediated का link. So adaptive and cell mediated humoral and cell mediated response, they are linked, and innate and adaptive response, they are also linked. किस तरह से? Complement fixation is a specific non-specific response, it is an innate response, but B cell आके this non-specific response को activate करता है. So essentially you have a link between innate and adaptive immunity. Is this clear? বুঝতে পেরেছো?
Yes, sir.
How everything is linked with each other? বুঝতে পেরেছো? How
Yes, sir.
everything, okay. Now let us say T helper cell, cell mediated T cell का role. I'll increase the size. So TH dendritic cells, this is a dendritic cell. This is also an antigen presenting cell, APC cell or antigen presenting cell. These cells are also killer cells. These are non-specific. How it is related with a specific T cell response. So what happens? They are class 2 MHC. They are since antigen presenting cells, immune cells. They will the antigen is presented over here. They are recognized by T helper cells. So dendritic cells or antigen presenting cells, they activate the T helper cells. So what they do is activated TH cells, they stimulate the dendritic cells to express additional co-stimulatory molecules like interleukins and cytokines. And what they do, they activate the cytotoxic T cell. इसको मार नहीं रहा है, लेकिन ये एक्टिवेट हो रहा है. दिस इज गेटिंग एक्टिवेट. ये जाके MHC1 वाला दूसरे टिश्यू, जो वायरस इन्फेक्टेड टिश्यू, जो कि यही है, इसी को दिस पर्टिकुलर एंटीजन को एक्सप्रेस कर रहा है, उसको जाके उसको मार देगा. So TH cells, they also help in humoral immunity. Activated B cell, ये जो T helper cell क्या किया? It is they have got MHC2. They are expressing processed antigen. Now this T cell helper cell will come and bind to this, and they will release interleukin 4 and other cytokines, and they will activate these particular B cells to release more antibody. Clear about this? How T helper cell will help B cell to release more antibodies. Clear?
Yes, sir.
Yes, sir.
And they will also express these antibodies also on their surface. So ये B cell भी जाके cell expressed antibodies है. So activated TH cells interact with B cells displaying antigen and produce cytokines that prompt B cell to mature and form antibody. So ये B cell mature हो जाता है, पक जाता है. पकने के बाद क्या होता है? They will start expressing more and more antibodies in the blood plasma. So there are steps of B cell activation. There are steps of B cell activation. We are not studying B cell activation. अभी नहीं पढ़ रहा हूँ, बाद में पढ़ाऊंगा. But basically, they activate the B cell with this interleukins, and then these activated B cells will then release antibody. So how a T cell and a B cell, humoral and cell mediated, they are linked through this T helper cells. TH cell क्या करता है? They produce cytokines, and that TC cells को activate कर देता है, and they recruit other WBCs and amplify innate other WBCs, मतलब न्यूट्रोफिल्स इन लोग को भी बुलाता है, इन्फ्लेमेशन करवा देता है. Now cytokine TC cells क्या करता है? Directly attack and kill cells with specific antigen. So TC cells are co-stimulated by TH cells and other cofactors. Co-stimulators. ये क्या करता है? ये बाइंड करके ये they release these perforins and ये होल बना देता है and the target cell will die. They will leak cytoplasm and they will die. Granzymes. TC cells bind to cells and release perforin and granzymes. And ये they make a hole. So और भी रहता है T regulatory cells, they release inhibitory cytokines, they suppress B cell and T cell activity. सब हो जाने के बाद तो बंद भी तो करना है, नहीं तो मारता ही रहेगा. तो कुछ सप्रेसर T सेल्स भी रहता है जो रोक देता है इन लोग को. तुम्हारा इंफेक्शन खत्म हो गया, हाँ इन्फ्लेमेशन को तो अभी तो कम हो जाना है, तो यू नीड दिस, ये भी हेल्प करता है. ऑर्गन ट्रांसप्लांट ये सब चीज नहीं है. सो नेक्स्ट है व्हाट वी विल बी डिस्कसिंग, वी विल टॉक अबाउट हाइपर सेंसिटिव रिएक्शंस. ठीक है. So I sort of end my class today. So just asking if you people have got any questions. Any questions? Last টা দেখতে পেয়েছিলে তো তোমরা?
Yes, sir.
Yes,
Okay.
sir. Yes, sir.
Okay. Any questions? हाँ राहुल बताओ क्या क्वेश्चन है? राहुल क्वेश्चन है? ओके 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 ठीक है. তাহলে কোনো কোয়েশ্চেন নেই আজকে?
No, sir.
ওকে. আমি তোমাদের নেক্সট ডেটা বলে দিচ্ছি, আরেকটা নেক্সট ডেটা অনলাইন নেব মোস্ট প্রবাবলি সানডে. তো নাফিসা সানডেতে তোমার পঙ্কজ স্যার থাকে না?
হ্যাঁ স্যার, সকালে 9:30
হ্যাঁ স্যাটারডেতেই প্ল্যান করার ইচ্ছে আছে. দেখি আমি তোমাদের জানিয়ে দিচ্ছি. ঠিক আছে.
Okay, sir.
ওকে স্যার।
আর পর্ণালী তোমার সৌরভ স্যার থাকে না সোমবার দিন?
Yes, sir.
আচ্ছা সৌরভ স্যার কটার থেকে?
হ্যাঁ স্যার.
সৌরভের তোমার প্র্যাকটিক্যালটা কটার থেকে সৌরভের?
স্যার 8টা থেকে থাকে।
8টার থেকে ঠিক আছে. আর সৌরভ তারপরে মানে করে দেবো.
স্যার বিকেল দিকে ক্লাসগুলো নিলে ভালো হয় অনলাইন।
বিকেলের দিকে অনলাইন গুলো নেবো, সকালে নেবো না. সকালে এই যে অফলাইন অনলাইন ভাবে এই দুটো করি, এটা থার্সডেটা আমার জন্যই রেখো তোমরা. হ্যাঁ অফলাইনে আসার চেষ্টা করো আস্তে আস্তে. অনলাইন যেদিন পারলে না অনলাইন গেলে, কিন্তু অফলাইনে এসে একটু সামনাসামনি কথাবার্তা জিজ্ঞেস করা, মুখগুলো একটু দেখতে হয় যে তোমাদের কতটা পড়া হয়েছে ঢুকেছে. একদিন না একদিন মুখ তো দেখতে হয় একটু. হয়তো অফলাইন পড়ানোটা একটু মানে অনলাইনেতে কাজ চলা যায়, কিন্তু সপ্তাহে বা মাঝে সাঝে একটু তোমাদের কতটা